communication across synapses. And we talked about the fact that when an action potential gets to synaptic end bulb, then it opens calcium channels. Calcium causes the release of neurotransmitter, and the neurotransmitters bind with receptors on the dendrite, and we changed this to a gradient potential. And what we did is we went through the neurotransmitters that are involved in the process. And then we looked at kind of two patterns for communication across synapses. One, the classic neurotransmitter that binds with the receptor. The other were neurotransmitters that bind with receptors that aren't channels, and they create an internal messenger called the G protein that activates a secondary messenger to control the response of the postsynaptic cell. Then we were looking at pathways by which neurons can interact, where you have one neuron eventually exciting a number of neurons, a diverging pathway. You have many neurons required to excite one neuron, a converging pathway. And then we talked about reverberating pathways that repeat themselves. And then another type of pattern similar to that, we have parallel pathways required to excite the postsynaptic neuron at the end. So I want to kind of use an example of these plus what we had talked about before to think about two patterns that exist within the central nervous system. So when we talked about summation in muscles, remember we used wave summation to create incomplete tetany and complete tetany. Okay? So the idea of summation is that you have repeated stimuli. And what we learned in muscles is that to go from incomplete tetany to complete tetany, we had to do what? Decrease the time between the stimuli, or the inverse would be increase the number of stimuli per unit time. Okay. So summation in this instance is going to be the same. And so what we're looking at is going back to the idea that on our dendrites and our cell bodies, we have a graded potential. And what we have to do is we have to create a threshold event at a trigger zone to get an action potential. And if we get an action potential, then we're going to have a excitatory postsynaptic potential. If we don't create a threshold event at the trigger zone, then the postsynaptic neuron wouldn't respond, and we call that a inhibitory postsynaptic potential. So an EPSP or an IPSP. So what we're going to do is take that idea and look at the way in which neurons can interact with each other. And we have two examples we're going to look at. One is called spatial summation. And the key to spatial summation is it's an example of a converging pathway where you have more than one presynaptic neuron that's interacting with a postsynaptic neuron. So the other example we're going to look at is called temporal summation, where you actually have one presynaptic neuron that actually stimulates a postsynaptic neuron many times. Right. So if we start over here, then A and B are presynaptic neurons. So they're going to release neurotransmitters from their synaptic end bulbs that are going to target the receptors on the dendrites. And so if neuron A is the only one that stimulates the postsynaptic neuron, neuron C, then we don't get a great enough graded potential. So we don't get a threshold event at a trigger zone, and we don't end up exciting the postsynaptic neuron. So the way we would have to make sure that the postsynaptic neuron is excited is we'd have to use more than one neuron to stimulate it. So we'd have to use both A and B. So that's what they're trying to show you here. So neuron A stimulates B, but we never reach threshold at the trigger zone, so nothing happens. Neuron B stimulates neuron C by itself, but we never reach threshold at the trigger zone, so we end up with an IPSP on the postsynaptic neuron. So what happens if neuron A and B both stimulate the postsynaptic neuron? Then we get a threshold event at a trigger zone, and we actually get an action potential on the postsynaptic neuron, or an EPSP. So that's the reason why we have those pathways that we just looked at, where we have many neurons that can interact with one neuron. Is because oftentimes it requires more than one neuron to stimulate the neuron to get postsynaptic potential. So in this example, what we have is we just have a linear relationship with a presynaptic neuron and a postsynaptic neuron. And instead of looking at space, which is more than one neuron over here, spatial, we're looking at how quickly the presynaptic neuron is stimulating the postsynaptic neuron. So if the presynaptic neuron stimulates the neuron once, and it doesn't reach threshold, and there's no stimulus for a little while, and then the second one, then we never reach a threshold effect at the trigger zone. So what the presynaptic neuron has to do is increases the release of neurotransmitters in time, so that we're getting a higher load of neurotransmitter release, so that essentially the presynaptic neuron is stimulating the postsynaptic neuron repeatedly very quickly. Then we'll get a buildup of neurotransmitters in the synaptic cleft, so that we actually get a threshold effect at the trigger zone itself. So this one is time, because we're looking at how fast neurotransmitters are being released. So temporal summation. And this one is spatial, because we're looking at more than one presynaptic neuron involved in the process. And the input, the end result is trying to elicit a action potential in a postsynaptic neuron. So just an example of tying those two things together we were just talking about. So what we do clinically is we put a bunch of electrodes on people's heads, and then what we do is we try to measure the depolarization events that are occurring within the brain itself. So we use electroencephalograms to try to determine when brain damage has occurred, and we also use electroencephalograms to determine whether somebody would be quote-unquote brain dead. So if we put an electroencephalogram on somebody who had tremendous head trauma and they're all flat lines, then we would consider the person brain dead, because there's no neurologic activity in the upper brain where we're actually picking up depolarization, repolarization events. So based upon the way in which we put the electrodes and the way in which the machine reads the electrodes, then we can look at different wave patterns, and they tell us something about the function of the brain. So alpha waves are one of the wave patterns that we would look at. And then just to quickly review the way that we interpret a wave pattern, is we interpret a wave pattern two ways. So anytime you're looking at a pattern that's a cyclic pattern like a wave, we look at how high the wave is. So that's amplitude. So one of the things we're going to see when we talk about this is the idea of amplitude, and that's how high the wave is. So you can be looking at waves that are simultaneous waves in the same time scale, and so one might be very low and one might be high. So we'd say this is a high amplitude wave compared to this one. So time would be the event going this way. And so the other thing we would look at is how many waves are occurring per unit time. So let's say this is a millisecond. Then how many waves are occurring in a millisecond? So the way this is expressed, the waves per unit time, is in kilohertz. And so that's what the Hz stands for, is kilohertz. So you've used this all your life, you just haven't thought about it. So when you get in your car and you adjust your radio to a different station, each station is transmitting with different waves per second. And that's what the numbers on the radio actually are the kilohertz or megahertz, depending on which frequency you're looking at. And so what's amazing to me is every cell phone has to have its own wave pattern. So ponder that one for a second. Yeah. So the more cell phones we have, the more different wave patterns we actually have to have. Otherwise, it would be a party wave. And you would call somebody and 8 or 9 phones would ring. So ponder that one for a little bit. It's pretty amazing to think about. Yeah. So anyway, alpha waves are 8 to 10 kilohertz. That's the pattern of waves per second. And they occur in normal physiology when a person is awake, resting, mind wandering, and eyes closed. And then are usually recorded in the parietal occipital region of the brain, so toward the back area of the brain. All right. So beta waves are faster waves, so they're more, almost double the frequency, so 14 to 30 kilohertz. And they normally become accentuated during mental activity and sensory stimulation. And so they're typically recorded on the frontal parietal regions of the brain. So why would parietal and sensory stimulation, why should that make sense to you? Because the parietal lobe of the brain is largely sensory, right? So it's largely where the sensations from your body go first and where cognition of those sensations actually occurs. So theta waves are fairly slow waves, so 4 to 10 kilohertz per second, so they're cycling very slow. And they're normal in children when drowsy and or sleeping as adults. And predominant theta waves in an awake adult suggest either tremendous emotional stress or brain or a couple of different brain disorders. So we use them diagnostically to try to figure out what's going on with people's brains. These are some of the wave patterns we also looked at in lighting tests. Where if you think it's wrong to lie, you'll be under emotional stress, and you'll get a phenomenon of theta waves. Okay. And then delta waves are really slow waves, but they're very high amplitude. So they are cycling very, very slow, but very, very high wave. So very slow and very high. And infants exhibit these waves when awake, and adults exhibit them in deep sleep. So it's kind of a cool pattern that what we know about children is that as the brain is maturating, we have many, many neurons, and then afterwards we get this slow loss of neurons over our life. And so we see behavior patterns change within the brain itself. If we get an increase in delta waves in an awake adult, it usually means serious brain damage. So that's one of the ones we would look at. Say if somebody's been in a car trauma and had a tremendous concussion event, then we would look at delta waves to try to figure out, how seriously the brain has been impacted.
Which one is REM sleep?
So, I tried to dig it out of the calculators. Yeah, but one of these is more dominant in REM sleep. My guess is theta waves. Yeah.
So in order from lowest frequency to high frequency, it's delta, beta, alpha, theta?
Yeah. I've been meaning to actually show you a printout on that. I just haven't had time to scan it or look what good yet. Okay, so yesterday we talked about the development of the brain in lab. So everybody good with that? And then we kind of went over different areas of the brain and from an anatomical standpoint. So we took a tour of the brain from the anatomical standpoint. So what I want to do is I want to finish talking about production and movement of cerebral spinal fluid. And then we'll go to lecture 3 where we actually go back and look at the structures we were looking at in the brain yesterday and begin to put function in it so that we begin to understand how different areas of the brain work. So one of the things that's critical is that when we supply blood to the brain and when we produce cerebral spinal fluid, we have a selective process that can limit certain toxins from getting into the brain and also at the same time limit pathogens that get into the brain so that we can get a really bad head cold where our nose runs like crazy, our eyes water, our respiratory system produces gobs of mucus, and noise is like crazy, but it doesn't affect brain function that could lead to death. And so there are only some pathogens that actually can pass through this process. So what we talked about earlier was that when we're producing cerebral spinal fluid and we do this exchange with the brain, we have what we call the blood-brain barrier. And so the blood-brain barrier is actually modified capillary beds that are lined with ependable cells, which are columnar-type epithelial cells. And so the ependable cells and astrocytes help produce cerebral spinal fluid. And so what we had yesterday in lab was a special name for that capillary bed that forms the blood-brain barrier, and that was the choroid plexus. Okay. So what we're going to find out is that within all of the ventricles of the brain, the two lateral ventricles, the third ventricle and the fourth ventricle, then we have this choroid plexus, and that's where cerebral spinal fluid is produced. And we produce cerebral spinal fluid at a reasonably constant rate in normal physiology. It can be influenced by elevated blood pressure, and it can be influenced by changes in blood volume, i.e. dehydration events in your body. Okay. So at any point in time, we usually have about 80 to 150 mLs of cerebral spinal fluid. So we now have this tremendous amount of cerebral spinal fluid. That's throughout the system. So that would be all the ventricles, the Cerebral Aqueduct that connects the third ventricle, the fourth ventricle, and the Subarachnoid Space. And we produce it at about 20 mLs per hour. So that over the course of a 24-hour day, we've produced about a half a liter, 480 milliliters of cerebral spinal fluid. So the inverse of producing it is, because we're constantly producing it, we have to constantly reabsorb it. And so we talked about in lab yesterday the blood vessel that sits in the longitudinal fissure that runs the total length of your brain on the superior side, which was the superior sagittal sinus. And then that created the little groove in the skull that we learned when we were learning the skull, which was the superior sagittal sulcus.
If you don't have enough cerebral spinal fluid, is it transfusible from one person to another?
Not that I'm aware of.
Okay.
What they would probably do is use medications to try to control blood pressure, change blood pressure. Right. So when we look at it, not surprisingly, it has a similar composition to blood, but it's not identical to blood composite of what we would find in the blood. So that tells us it's a selective process through the blood-brain barrier. So we would find things that we would think would be important. So glucose would make sense because glucose is our preferred method of making ATP. And the brain is a high consumer of ATP. So the brain is a high consumer of oxygen because of the mitochondria in the brain itself. So we have proteins in our blood that provide a number of different functions. But when we get to the cardiovascular system in the next quarter, one of the functions we're going to talk about with blood proteins is they create osmotic balance between our tissues. And so dissolved proteins are critical in kind of keep an isotonic situation between your tissues and the fluid around them. So it's not surprising we have proteins in cerebral spinal fluid as well, because there's such an important osmotic component to to helping us maintain water balance, so our brain doesn't swell and swell and swell and swell from edema. And then the place we would see that more frequently would be in high elevation problems, particularly climbers like on Everest and stuff, can get into serious trouble because as they get up there, a number of factors are influencing their body, and they'll suffer from edema. And so the most two common types of edema are pulmonary edema and cerebral edema. So I've seen a couple of pictures of climbers where their eyes are completely out of their sockets almost because of the pressure of the brain behind them. And so it leads to death. So the climbers die from it. And then lactic acid is a byproduct of anaerobic respiration in muscles. So it's the end product that muscles need to do to free up NAD so they can continue to do glycolysis. Well, not surprisingly, certain organs, your brain, your liver, your heart, can actually run lactic acid through the Krebs cycle and make ATP out of it. So it's kind of an interesting pattern that we would see. So the amount of lactic acid would be variable based upon what activity the person has been actually being engaged in, on how anaerobic they were. Okay. So if it was at the end of a 26-mile marathon, then we would see quite a bit of lactic acid compared to somebody who was sleeping. So very.
Is that one of the reasons why you like a runner's high after you get done running?
No, that's actually those endorphins we talked about.
Oh,
That you get that release of endorphins, yeah. It gives you that runner's high. And then urea, because that's a waste product that's created because we're breaking down proteins. So, and then nitrogen is pretty toxic to us. So we use urea as a waste product to actually get rid of the breakdown of those proteins. And then we have critical cations, or positively charged ions. And the ones we've talked about, sodium, potassium, and calcium, would be fundamentally important for brain function. So sodium would be important because it helps us do what? Create what membrane phase? Movement of sodium into the neuron causes depolarization. So if we're going to have neurons that can undergo an action potential, they have to be able to undergo depolarization. So now why would potassium be important? Because it's required for repolarization. Why would calcium be important? It's required for the release of neurotransmitters at synapses, right? Because an influx of calcium is what triggers the release of neurotransmitters. So those three definitely make sense. And then magnesium is one of our body's more important coenzymes. So we produce proteins that facilitate reactions that allow us to do things at a rate that would not normally occur. But we also want to be able to control the activity of these enzymes. So one way that we can do it is we can make the enzyme in a non-functional form. When we combine a coenzyme with it that activates the enzyme. So we can control where the enzymes are active. So we're going to spend quite a bit of time looking at that when we do metabolism in the next quarter. But magnesium is a common coenzyme that we use. So negative ions, antions, chloride always likes to hang around with sodium. And then bicarbonate, which is the HCO3, is an incredibly important buffer to your body. And so what do buffers do? They help maintain pH. So our blood, changes in blood pH are real problematic, and we'll do it in the next unit. But we don't like our blood to change one pH unit without having some consequences to it. And so we have to have buffers that allow us to maintain a reasonably constant internal pH. And bicarbonate is one of our most important buffers. So it's obviously important in cerebral spinal fluid because it's going to help maintain pH of the situation. So we produce cerebral spinal fluid for chemical protection, these ions. So if we're low in sodium in a clinical environment, we would say that you were in a state of hyponatremia. And people in severe cases of hyponatremia will become disoriented. They'll lose the ability to think effectively. They won't be able to answer questions you ask them. They could go into a coma and die. And so it's very important that we maintain sodium levels. There was actually a marathon runner 5 years ago in Hawaii who showed up at an aid station. She was totally confused. They started asking her questions, and she was totally confused. They gave her water to drink because they thought she was dehydrated. But unfortunately, she was hyponatremic, so as soon as they gave her water, it made her more hyponatremic, and she went into a coma and died. Yeah. So certainly from a standpoint of working with people who are exercising, this hyponatremia is a critical issue that leads to serious problems. So there's kind of a cool historic perspective to that. So there was a university where the football players, when they came back in August and started working out really hard, would get really ill, and a couple of them actually died. So it was actually a physiology instructor at the university who concocted this drink that the football players would drink to prevent them from entering hyponatremia. And it happened to be the University of Florida. And the drink became known as Gatorade. Yeah. So pretty amazing. So the inverse would be hypernatremia, and that leads to mental confusion as well. So my wife had a patient who had been coming to her for almost the whole 21 years that my wife had been in practice here in town just a little while ago, and she was an elderly woman. And one of the things that had happened in the year and a half preceding this visit was that her husband had passed away. And so because her husband had passed away, the only reason she was cooking was for him. So she just quit cooking, and so she just started eating a lot of canned soups, like Campbell's soup and stuff. And my wife noticed when she came in that she was really much more confused than she'd ever been historically. So started doing an analysis. And one of the things that was elevated was her blood calcium level, her blood sodium levels were incredibly high. And so she was actually in a state of hypernatremia because of the high sodium load she was getting from the pre-made soup. So it's very interesting. And it helps circulate all these things around. So when we look, then this is kind of a review of what we did in the lab yesterday, which is the fact that our brain and spinal cord form as a tube. And then as the brain enlarges, we develop those primary vesicles and then secondary vesicles that we talked about. But the end product is we have spaces inside of our brain because our brain forms as a tube. So in the telencephalon, which forms what major area of the brain? Telencephalon.
That's the very covering.
The cerebral cortex, right? So the cerebral hemispheres. Then we have a lateral ventricle in each one. So we have a lateral ventricle in the left cerebral hemisphere and a lateral ventricle in the right cerebral hemisphere. And these ventricles will actually have choroid plexus where they produce cerebral spinal fluid. So there's a connection between each lateral ventricle and the third ventricle, which is associated with what secondary brain development area? So associated with the thalamus, the hypothalamus, the diencephalon. So it's called the third ventricle. And so what interconnects the two lateral ventricles and the third ventricle is an interventricular foramen. And so that allows fluid being produced in the lateral ventricles to move into the third ventricle. And the third ventricle also has a choroid plexus in it. So it actually produces cerebral spinal fluid. So what we have is a duct called the Cerebral Aqueduct that connects the third ventricle and the fourth ventricle. And the Cerebral Aqueduct was a landmark for what area of the brain? So it was the only area that retains the same name from primary to secondary. So the mesencephalon, or what we would call the midbrain, generically. And so the cerebral aqueduct passes through the mesencephalon. It connects the third ventricle to the fourth ventricle. The fourth ventricle is associated with the pons and the medulla. So the pons arises from what primary brain vessel? From the rhombencephalon. Then remember, the rhombencephalon divides into metencephalon and myelencephalon. So the pons arises from the metencephalon, and the medulla oblongata arises from the myelencephalon. Right. And they are associated with the fourth ventricle. So from the fourth ventricle, what the fluid is going to do is it's going to exit the internal compartment within the brain and go out into the subarachnoid space. So we have an opening that's just inferior to the cerebellum that's midline. So since it's midline, it's called the median aperture. And then we have two that come out the sides of the fourth ventricle, and those are called lateral apertures. And that takes it all out into the subarachnoid space. So this is just a diagram that gives you a visual interpretation of everything that we just said. So all ventricles have choroid plexes. So fluid that's produced in the lateral choroid plexes has to eventually exit the lateral and go into the third. So the holes they pass through are the interventricular foramen. Sorry about that. And then the fluid that's produced in the third ventricle and all the fluid that was produced in the lateral ventricles has to exit the third ventricle and go into the fourth ventricle via the cerebral aqueduct. And then fluid is also produced in the fourth ventricle. So fluid from both lateral ventricles, fluid from the third ventricle, fluid from the fourth ventricle, exits the fourth ventricle via the median aperture and the two lateral apertures. Now, what we're going to talk about in lab tomorrow is the fact that your spinal cord has an open space in it all the way down through the spinal cord to the bottom, which is again because the spinal cord is used as a tube. And so that space that we have right here in our spinal cord going all the way down is called the central canal. So that's the other place that it can actually go from the fourth ventricle, because it can actually go down the center of the spinal cord all the way down and through the central canal. So if you wanted, you could actually make an arrow off the fourth ventricle, right central canal, and then point it toward the subarachnoid space, and then that would fill that gap in for you on this flow chart. Then it also can go down the central canal of the spinal cord. And so what then happens is all the fluid's going to be in the subarachnoid space. And then it moves in a predictable pattern, which has allowed us to use a diagnostic test to try to figure out what's going on when people are having significant issues with brain function. And it's called a spinal tap. And so what happens is the spinal cord actually terminates before the lumbar vertebrae. And so we can actually use a big bore needle and go through all the soft tissue to the duramater. And then inside of that big bore needle, we can use a fine bore needle to penetrate the duramater or arachnoid mater and go into the subarachnoid space. And then we can withdraw cerebral spinal fluid and do tests on it. And so we do it in the lumbar region because the spinal cord terminates, so we can't damage the spinal cord with the needle. But the pattern that exists that allows us to do it is the fact that cerebral spinal fluid will travel down the posterior aspect of the spinal cord and go around and go up the anterior aspect of the spinal cord. So we can actually collect cerebral spinal fluid from the lumbar region. We know it was inside the brain and has traveled down. And that allows us to actually then try to figure out what was going on within the brain itself, even though we're a ways from the brain itself. And then what happens is all the fluid then eventually goes up and over the brain and is going to be returned to the blood in that superior sagittal sinus, which is a blood vessel we're seeing at the top of the picture. So in the next picture, all I'm going to do is I'm going to rotate the brain for you. So we're looking at a mid-sagittal view here. So in the next picture, what we're going to do is we're going to rotate the brain so we're looking at a frontal view of the brain. And so the frontal view of the brain gives us a better visual of how the lateral ventricles are connected to the third ventricle, where the third ventricle is relative to the fourth ventricle. So the two at the top that kind of look like the rabbit ears are the lateral ventricles. And then the lateral ventricles are going to be connected to the third ventricle via those interventricular foramen. And then the third ventricle is going to be connected to the fourth ventricle via the Cerebral Aqueduct. And then again, from the fourth ventricle, we can go out the lateral armatures, median armature, or down the spinal cord. And then what happens is it flows over the brain in the subarachnoid space. And now at the top, we can see how that blood vessel, the superior sagittal sinus, sits in the longitudinal fissure of the brain. And it's actually surrounded by the duramater. So the duramater actually encapsulates that blood vessel. So one of the things we're going to do tomorrow is try to catch up with what we didn't get done yesterday, which was the sheep brain dissection. So when you first get your sheep brain, if you look at the top center of the brain, you can actually see this blood vessel sitting in the duramater of the sheep brain. And so what we're going to do now is we're going to just enlarge that area where the superior sagittal sinus is sitting and look at it. So we're going to actually just enlarge this area right here in the next image so that we can see that. So here's our two hemispheres. So in this picture, the green is the duramater. The purple on the inside of the green is the arachnoid mater. You can see the fiber pattern of the arachnoid mater, which is where it got its name, because it looks like little spike rays. So the little space between the green and the kind of orange or reddish is the pia mater. So this space is the subarachnoid space. So as the fluid is passing through the subarachnoid space, what happens is we have extensions of the arachnoid mater into that blood vessel at the top, the superior sagittal sinus. And so what we end up with then is these little extensions that we call villi. So villi are upward finger-like folds. And so these villi extend into the blood vessel. So the fluid is actually in the subarachnoid space at this point, and then it re-enters the blood vessel. So ideally, if we're going to make 20 mLs per hour, 480 mLs per day, then at the other end we need to be reabsorbing 480 mLs per day, so that production equals reabsorption. So one of the things that happens clinically if you do a spinal tap and you withdraw fluid is the person now has less cerebral spinal fluid. And one of the consequences is some people get some significant headaches from that, because now the brain is actually sitting on the floor of the cranium because cerebral spinal fluid was withdrawn and it's no longer buoyant. And it takes a day or two for them to recover the amount of cerebral spinal fluid that was actually taken out when they did that. So this is actually a child with what we call hydrocephalus. And so in today's world, at least for the part of the world that we're usually more cognizant of, which is one where healthcare is reasonably affordable and available to people. And we don't see babies at this extent of the development of this. But if you go to underserved areas of the world, you can still see babies in this position. And so what's happened is there's actually a blockage of flow so that the fluid in the lateral ventricles isn't allowed to move like it's supposed to move. And so this light area is the lateral ventricle on this side. And this light area is the lateral ventricle on this side. So we can see that this lateral ventricle is almost twice the size as this lateral ventricle. So what's actually happening is cerebral spinal fluid is being retained in the spaces inside the brain. It's causing the brain to swell. So we have cerebral edema going on. And then what's actually happening is because the baby's skull doesn't have sutures yet, then the baby's skull bones are being pushed further and further apart. And the top of their head can get bigger and bigger and bigger. And usually it leads to their death unless you can resolve this. So in today's world, because we have all these well baby checks that people do, we can pick up changes in behavior patterns and problems in developmental patterns neurologically more quickly, and then we can figure this out. And what they do is they can put shunts in to move fluid so it doesn't actually build up. So when my wife was first out of residency, she was part of a practice in rural Wyoming. And because she had gone to a residency in a major hospital, she had actually encountered this in the baby, and they caught it early and stuff. So one of the babies that and at the time she was delivering babies, so one of the babies she delivered, when they brought her back for a 2-week check, had neurologic problems, and she quickly did some tests, figured out the baby was having blockage of flow of cerebral spinal fluid, and then they life flighted the baby from this rural area of Wyoming to Salt Lake City where they could do the surgery. And so the baby didn't end up like this. There's actually, if you went out on the web and you put Crypts of Palermo in, so Palermo is a city in Italy, and there are these crypts under all these Catholic churches, which are underground passageways. And a lot of these crypts have a lot of human bone material as the artwork on the walls. And there's a couple of pictures where if you look closely at them, there's a number of baby skulls, and a lot of the baby skulls, top of their heads are gone. And these were all babies that died from hydrocephalus back centuries ago. And what used to happen is when the baby's head started swelling like that, the family didn't know what to do, so they would bring the baby to the convent where the nuns were, and then the nuns took care of the baby until it died. So there's some research been done on burial grounds with those Catholic churches showing a fairly high amount of babies with hydrocephalus that were there.
Isn't genetic movement to any certain?
I think it's fairly multifactorial. There's not the easiest thing in medicine is that one-to-one kind of smoking gun.
Yeah.
But unfortunately, most of medicine doesn't work with that principle. So there's many factors that can influence. But I haven't read if there's any familial pattern. And I haven't looked to see if there's anything in the pattern of EEG. All right, so now what we're going to do is we're going to kind of begin to think about what different areas of the brain does. And so this is going to be an overview and not a detailed analysis of each area of the brain. Because if you went to University of Washington or Washington State, where they teach neuroanatomy and neurophysiology, the textbook is as thick as your textbook. So we're just going to gloss over an overview. All right. So all we're going to do is we're going to start at the base of the brainstem, and we're going to work our way up through the brainstem into the higher brain. So part of what we're going to do is think about the brain and the spinal cord as part of the central nervous system, being the conduit through which sensory information from your body, the peripheral nervous system, enters the central nervous system, is conveyed to the appropriate areas of the brain, largely the parietal lobe of the brain, where cognition of those sensory events occurs. And then the brain determines a response to that. And so what we're going to see is a pattern where motor activity arising in the brain passes down through the spinal cord to go out to the peripheral nervous system. So what we're going to see is some unique responsibilities to each area of the brain. And then the brain and central nervous system's organization that allows for this communication from your body to your higher brain centers, and then from your higher brain centers back out to your body to create a pattern that keeps us in homeostasis. Okay? So when we look at the medulla oblongata, which is the bottom of our brainstem, then one of its very critical roles is that it has your cardiovascular system. So if you'll recall, when we did myocardium or cardiac muscle, one of the things we said was that cardiac muscle was involuntary. So we don't consciously have to think about making our heart beat. When we actually get to our heart, what we're going to find out is the heart doesn't need your brain at all to create a steady pattern of contraction and relaxation, because the heart has its own pacemaker system. So the heart itself can control a reasonably steady rate of contraction, relaxation. So what our brain has to do is communicate with our heart so that if the internal environment in your body is changing because you're increasing your activity level, then we have to be able to accelerate your heart rate based upon delivering oxygen to skeletal muscle cells that are using more oxygen, and your brain is using more oxygen. Then when you're done with that activity, it has to bring the heart rate back to normal. So what the cardiovascular system does is it overrides that normal pattern that the heart creates itself and allows your heart rate to accelerate under exercise and then be brought back down to its normal level after you're done with exercise. So that's the real importance of the cardiovascular system, and that it regulates rate and force of contraction, particularly based upon your body's need for oxygen, carbon dioxide exchange. And then it also helps control the diameter of blood vessels. So what we can do by changing blood vessel diameter is we can control blood flow to extremities, and we can control blood pressure. And so we can use blood vessels to do both of that. So if you become really, really cold, what happens to your hands? They get cold because you want to maintain the core temperature of your body. And if you're putting a lot of blood out into your extremities, the blood is cooling, and then you're going to continue to cool your core temperature. So you begin to shut your extremities down so that you don't have as much blood flow out there, so you can try to maintain core temperature of your body. So those are the kind of things that the cardiovascular system is involved in. Okay. And then near the cardiovascular system, there's a medullary rhythmicity area. And it controls your basic pattern of respiration. So your respiratory rate. And that would make sense because the role of the heart is to pump blood to your lungs to give off carbon dioxide and take on oxygen. So you want to be able to control the rate of that pumping of the heart because it's going to influence the carbon dioxide load and the oxygen load in your blood. And you also want to elevate your change in respiration as well. So as you begin to exercise, not only does your heart rate go up, but your respiratory rate goes up. All right. So what the medullary rhythmicity area does is adjust your basic rhythm of breathing. And so it has two areas, an inspiratory area that makes you breathe in, and an expiratory area that makes you breathe out. And what we're going to see in normal respiration is the only active area is the inspiratory area, because breathing out is a passive event, because we just stop the diaphragm from contracting, and pressure is what works to move air. Right? So typically we only use the inspiratory area. As we move from quiet breathing to labored breathing like an exercise, then we have to engage the expiratory area to help us expire air. And in things that are modifications of breathing, such as vomiting, coughing, and sneezing, are also done by the medulla. So that's one of the reasons why somebody can show up in a clinical environment with a tremendous head trauma from a car wreck or something. And we can use an electroencephalogram and it shows no brain function in terms of the upper parts of the brain, but their heart is still beating and they still have a basic respiratory rate, as long as the medulla has been unaffected. But you can also have somebody break their neck where the medulla is traumatized and they die quickly because these two centers have been impacted. All right, so the other thing that the brainstem function is a conduit or a connection between your spinal cord and your higher brain center. So all sensory information coming through your body, entering the spinal cord, has to pass through the medulla to get to the parietal lobe of the brain, where cognition is going to occur. And then our motor cortex is on our frontal lobe that we talked about yesterday. So any signals that you want to send from your brain to skeletal muscle to accomplish a task using skeletal muscle has to pass through the medulla as well. And so the medulla is a critically important conduit for flow of impulses toward the brain and away from the brain. And what we see on the medulla itself is a kind of cool pattern. It's called a pyramid. And so the nerve fibers, axons that are in the pyramid, are arising from neurons on the primary motor area of the brain, on our frontal lobe. And then what happens is they pass down through the brain, and in the medulla they cross. So that actually when we are contracting our left hand, it's actually the right side of our brain doing it. Because these cross in this pyramid. So it's kind of one of the cool patterns where we've got this crossing pattern that occurs within the pyramid for these motor tracts. So that the opposite side of the brain is actually controlling the muscles on the opposite side of the body. So then if you have a patient who's had a stroke on the right side of their frontal lobe, the paralysis we would see from the stroke would be on the left side. All right. So, and then what was a nucleus? We talked about it when we first started doing this. When we're dealing with the central nervous system. A group of neuron cell bodies in what we consider kind of white matter areas of the spinal cord. So where all these tracts would be white matter. And so what happens is, as information is coming into your brain, into your spinal cord from your body, and goes up to the brain, some of the neurons that started in the spinal cord are actually going to terminate in the medulla, and then new neurons are going to carry the information on out. So these nuclei, the nucleus gracilis and the nucleus cuneatus, are where cell bodies are located, and that's where neurons who originated in the spinal cord and carried information up, they're going to synapse with new neurons that have to carry it on up into the brain. Okay? So that's what these nuclei are. They're sensory relay centers. And we're going to kind of put that together in a pattern for it. Okay. So we're going to do this in lab tomorrow. We're going to learn the anatomy of the spinal cord. But one of the critical roles of the spinal cord is to carry sensory information from your body to your brain, and carry motor information from your brain to your body. So when we see spinal cord trauma, where the spinal cord has been severed because of a car wreck or some event, then we have paralysis because the motor information cannot go past the damaged area of the spinal cord. And we see a loss of sensations because the sensory information can't arise above the damage to the spinal cord. Okay? So what we do is information carrying sensory information on this is all blue. So these are all what we would call tracts. They're conveying discrete types of sensory information. So since the information is arising in the spinal cord to go up to the parietal lobe of the brain, then we say they are ascending tracts. And then the beauty of the way in which these tracts are named, and we're not going to get into the detail of what each tract does, but the beauty of it is because the neurons arise in the spinal cord and travel upward, then all of their names start with spinal, 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 spinal. So all ascending tracts are going to start with spinal because the neurons arose in the gray matter of the spinal cord. And then they're going to terminate in the brain somewhere. So they're all named from the fact that they started in the spinal cord and they're terminating somewhere in the brain. So spinal thalamic. So where is this going to terminate? In the thalamus. So spinal thalamic, spinal cerebellar. So where is this going to terminate? In the cerebellum. There's a cool pattern to the way the ascending tracts are named. Okay? So what we're trying to do is we're trying to put together kind of a communication pathway between your body, the spinal cord, and the brain, so that we have sensory information that terminates in an interpretive area where we create cognition, which is the awareness of whatever sensory information it was. So what we do is we call the neuron that's part of the peripheral nervous system nerve, whose cell body is in the dorsal root ganglion, the first order neuron. And it's going to arise from some type of receptor out in your muscle or your skin. And then it's going to synapse with a neuron whose cell body is in the part of the spinal cord gray matter that we call the dorsal horn. So it's the dorsal part of the butterfly. These neuron cell bodies have axons that are going to exit. In this example, it's actually going to cross the spinal cord and then become part of one of those tracts, spinal thalamic tracts. And then the axon is going to actually travel through the medulla, through the midbrain, and go all the way to the thalamus of the brain. And then in the thalamus of the, and so it's a second order neuron because it's the second neuron in the pathway. And then it's going to synapse with the neuron whose cell body is in the thalamus. And then the role of the thalamus is to relay it to the appropriate area on the primary somatosensory gyrus so that we have cognition of whatever sense it is. So the neuron who arises in the thalamus and goes up to the somatosensory area is a third order neuron. So the pathway required for you to stimulate a receptor in your finger and have that end up in your brain requires 3 neurons. And it requires 3 neurons that communicate with each other. But think about this, if you put your finger on something really hot, how quickly you know it's hot. And how quickly that cognition of pain arises. So it requires 3 neurons to carry it. Okay. Then what's amazing is we've actually mapped out the primary somatosensory area. So if it was my index finger that I put on something really hot, that pathway would carry the information right here to this area right here, where our index finger is. So this is actually that post-central gyrus on the parietal lobe of the brain, and that's where all the sensations arise. So that's where cognition would occur. So it's pretty cool. Think about it. So you don't have to memorize all of So ultimately, if we're stuck our finger on something really hot, then we want that sensory information to go up, the cognition of damn that hurts, so that you will create a motor response to move your finger before you cook it and destroy your finger. So what has to happen is, once the cognition arises that your finger is in trouble, you got to create a motor response. So the motor response is going to be initiated on the precentral gyrus, on the frontal lobe of the brain. So you're going to have a neuron cell body in the gray matter on the surface of the brain. So the reason why the cerebral cortex is highly folded with gyrus and sulci is to increase the amount of gray matter on your brain. And so what we're going to do then is have a cell body who's in the gray matter on the cerebral cortex, whose axon is going to pass down through the hemispheres, pass through the cerebral peduncle to get into the brainstem, pass down through the brainstem into the spinal cord, and then eventually it's going to synapse with the neuron whose cell body is in the ventral horn of the spinal cord. So this anterior portion of the gray matter of the spinal cord. And then its axon is going to exit and go out and become part of a peripheral nerve. That's eventually going to end up at a motor unit in a skeletal muscle that's going to make it contract. So since we number the sensory neurons, first order, second order, third order, then motor neurons are done by using upper neurons and lower neurons. So our upper motor neuron cell body is going to be in the brain, and its axon is going to travel down your spinal cord. So think about this: if it's going to go to a muscle in your foot, then that neuron cell body is in your brain, and its axon is going to go down to the lower part of your spinal cord. And that cell is that long, transversing all of the central nervous system. And then it's going to synapse with a new neuron whose cell body is in the ventral gray horn of the spinal cord. And then that axon is going to travel down a nerve like your sciatic nerve and go all the way to your foot. So the second neuron is also a very long cell. That's what amazes me about the nervous system, is that scope of distance that a cell is spanning. It's just absolutely amazing. Yeah. And then all along the way, they have cells that are association cells that help support it, right? So in the central nervous system, the myelin would be created by oligodendrocytes. And in the PNS, out here in the muscle, the myelin would be created by Schwann cells. So we have these cells that support it as it transverses this distance. It's amazing. Right.
Is there a condition whereby these cells would experience a form of hypertrophy?
Yeah.
Okay, so they would get bigger from...
Yeah, and less function.
Okay.
So when we look at the spinal cord, so on this picture, the red are what we call motor tracts, and they're descending because they're rising in the brain and going down. Now, so you don't get confused, the spinal cord is bilaterally symmetric, so the blue would also be over here and the red would also be over here, but they just separated so it was easier to see them, right? So I didn't want you to go away thinking that one side of the spinal cord is sensory and the other side is motor. So we're bilaterally symmetric, so we're both of that. Now what's amazing is the motor tracts are all going to be named from where they start in the brain. Cortico for cerebral cortex, rubro for the red nucleus, reticulo from the reticular activation system, and vestibulo from the vestibular area. But they're all going to end in spinal, because they're all going to terminate in the spinal cord. So there's a cool pattern to it. Okay. And then again, we've actually mapped the primary motor cortex as well. So we know, for example, that the finger, the muscles that would allow us to move our index finger are right here. So we know exactly where those are as well through some experimentation. All right. So the other thing that occurs with the medulla is nerves that arise from the brain and pass out into the facial area of the neck arise from the medulla. So nerves that arise from the brain are called cranial nerves, and nerves that arise from the spinal cord are called spinal nerves. And yesterday we worked on 12 cranial nerves. And what we discovered was that nerve 1 was associated with the cerebral hemispheres, and as we worked our way down the numbers, that the larger numbers were associated with the brainstem. So we were looking at the medulla. We had a number of nerves that arise from the medulla area. So at the medulla pons interface, we find the vestibular cochlear. On the side of the medulla, we find the glossopharyngeal, we find the vagus, and then on the anterior aspect of the medulla, we find the hypoglossal. So it's not surprising that those nerves that when we're looking at them on the brain that were associated with the medulla are arising from the medulla. So the vestibular cochlear nerve actually has two components to it. The word cochlear is a reference to the cochlea, which is involved in creating the sensations we need for auditory interpretation or hearing. The prefix vestibulo is coming from the vestibular apparatus, which is equilibrium. And so this nerve is a sensory-only nerve that's receiving incoming information from your ear for auditory or hearing, and incoming information from the other part of your ear, which is involved in equilibrium. So the part that's associated with the medulla is the part we use for hearing. So sensory receptors for the cochlea itself. So glossopharyngeal, so pharyngeal is your throat, pharynx. Glozzo is where did we see that when we did muscles? Tongue. So the muscle tells us where it's going. So we go to our throat and our tongue. So the glossopharyngeal is both sensory and motor. So it has sensory information coming in, motor responses out. But this is sensory only. Okay. And so the glossopharyngeal is going to be involved in taste, in swallowing, and salivation. So production of saliva. So what we can kind of do is we can kind of draw a generic head and neck. And what we can kind of do is begin to put structures on it as we're doing this. So this would be your mouth. And inside your mouth you have a tongue. And so what we can do with the tongue is we can divide the tongue into the anterior 2/3 and the posterior 1/3. And the glossopharyngeal does the posterior 1/3 of your tongue for taste. Okay? So that's the taste. And then the glossopharyngeal would also be down here in our neck because it's involved in swallowing. Okay? And then also associated with our mouth because of salivary glands in our mouth. Right? So now we put ears on our guy. And so our earrings would be eight because the clear part is involved with your ear, right? So nerve 10 was the vagus nerve. Now the vagus nerve is weird because we have to put the rest of the body in place. And so we can have our heart and our lungs and our diaphragm and our stomach and stuff down here. So the vagus nerve goes to your heart, your lungs, your digestive system. So the vagus nerve is your weirdest cranial nerve, does nothing in your head. It goes down into your organs. So it's the weirdest one. So it has both sensory and motor impulses from the viscera.
So on number one, we need to cross out the motor impulses?
Well, what it does, what I intended to do, which I didn't do well, cross it out. Because what I intended to do is, we use hearing as a very important way to respond to the environment around us. But the information coming in is sensory only. The motor response is not done by the vestibular cochlear, so I didn't mean to confuse you with that. So the spinal accessory is going to control impulses for swallowing. So where are we going to put nerve 11? So nerve 11 is actually going to be in your neck. And it also does a muscle that comes across here, and a muscle that goes right here. So your trapezius and your sternocleidomastoid. So 11 not only does swallowing internally, but it does these muscles externally in our neck as well. Okay? So those arise all off of the medulla oblongata. So as we move up from the medulla, then we encounter the pons. And the pons continues to act as a conduit. So anything going through the medulla that's going to go to the brain also has to go through the pons. And anything arising in the brain going to the spinal cord also has to go through the pons. So the pons is still an important bridge connecting the parts of the central nervous system. But also, on the back of the pons and medulla, we have a structure called the cerebellum. And what we're going to see is the cerebellum is critically important to coordinating muscle activity. So not only do you have to have longitudinal fibers going up and down through the medulla, but you also have to have transverse fibers coming from the pons and going into the cerebellum. So you have transverse fibers that connect to the cerebellum. You have these longitudinal fibers that we've talked about in the past. And then what we also have in the pons is two respiratory areas. And they're going to interact with the one in the medulla. So remember, the one in the medulla, the medullary rhythmicity area, controls your basic respiratory pattern. So when you're quiet, the pattern of breathing. But when you exercise, you have to be able to accelerate your breathing. And then when you're done exercising, you have to be able to bring your breathing back to normal. So it's actually the pons that allows you to adjust your respiratory patterns. And so one of the areas in the pons is the pneumotaxic area. So the first part of this, pneumonia, pneumotaxic, so that's the way to remember it. So it transmits inhibitory impulses to the inspiratory area. So if it's creating IPSPs in the inspiratory area, would it slow respiration down or speed it up? It'll slow it down, yes. So it actually inhibits respiratory patterns, brings it back down after you've been exercising. And then the other area is called the apneustic area, and it transmits stimulatory patterns. So it would do what to respiratory patterns? It would accelerate them, right? Okay. And then the pons also contains the nuclei of 4 cranial nerves, the trigeminal. And so what we're going to see, the trigeminal gets its name because it has 3 main branches. Hence the word tri. And one of the branches is going to go to our mandible. The other branch is going to go to our maxilla. And then one branch is going to go to our eye area. So if we now put more figures on here.
Did you skip 12?
Yeah,
Yeah, what version was 12?
Oh, hypoglossal idea. So that one's pretty straightforward. Where's it going to go? Under our tongue. So our hypoglossal is going to go under our tongue. And in 5, because we have ophthalmic glands, maxillary glands, mandibular glands, then 5 is going to go to all 3 areas of our face. Then we're going to work on that tomorrow in the lab, what areas we want. So our trigeminal was nerve 5. So our abducens is a weird little nerve. So what's the first part of this word if I just did this? Or let's do that.
Abduct.
Abduct. And so what was the word abduct? A reference to
Bringing away from the bone.
bringing away from the main line. So we have a muscle we learned on our eye, the lateral rectus. And when the lateral rectus contracts, it moves the pupil toward the side of your face. So it abducts the pupil. And that's exactly what the abducens nerve does, is it innervates the lateral rectus. So we have 6s on the lateral aspect of our eyes, or the lateral rectus. So is that cool? Pardon? Yeah. So facial. So where's your face? All of this stuff, right? So your facial nerve does saliva, tears, all those muscles of facial expression. So the facial was nerve what? 7. So 7 also has to do with the face. Some guys getting a little crowded there. Right? And vestibular cochlear, remember, which is nerve 8, we had one which is a cochlear component, which is hearing. So the vestibular component is for balance and equilibrium. So if somebody has vertigo, in really bad cases, they cannot even stand up because their head is going in circles. And that's the vestibular cochlear information. And just okay, so as we move upward from the mandible to the pons, and our next area is the midbrain, which we can also call the mesencephalon, right? And on the mesencephalon, not surprisingly, we have the continuation of those ascending sensory tracts and descending motor tracts. And the midbrain was critical in that it provides the bridge between the brainstem and the cerebral cortex. And that bridge was called the Cerebral Peduncle. So the cerebral peduncles are associated with the midbrain. We're going to see them in this little piece right here, connecting those ascending sensory tracts and descending motor tracts from the thalamus and up. And then we had on the back of the midbrain 4 bumps, so we call those the corpora quadrigemini. We had 2 upper bumps, which were the superior colliculi, and 2 lower bumps, which were the inferior colliculi. And so the superior colliculi are reflexes related to visual stimuli. So that's kind of fun. You could test this on someone. If you kind of made them set with the ruler that was held like this, and you flashed the light over here, then they would turn quickly to see what the light would be, and that would be your superior colliculi in action. So the response of your head, neck, and upper body to visual stimuli. So very protective with peripheral vision. Hear a growl over here from a light. Right. And then the inferior colliculi are reflexes related to auditory stimuli. So this one's great. If you have somebody sitting in a chair, they don't know you're back there, and you pop a balloon or something. in action. Okay? And I'm out of time, so we're going to finish this on Thursday. So what we're going to do on Thursday is we're going to finish this